and Brooke graduated in 2017 from Coastal Carolina University. So both are non-Mainers living in Maine now, and we're very excited to have them. So let's give a round of applause for our guests today.
Well, thank you. Can everyone hear me? Yeah. Awesome. Thank you for that introduction, and we would also like to start off by thanking you all for allowing us to participate in your Earth Week celebration. Like Ms. Ferrante said, we are both master's students at the University of New England, and today we're going to talk a little bit about the shark research that we do right in our backyard in the Gulf of Maine. So going to a little more of an introduction to ourselves. Once again, I am Brooke. I grew up in rural Maryland, but I kind of got my interest in marine science by spending a lot of time in the Florida Keys where my grandmother lived. And this led me to go to an undergraduate program at Coastal Carolina University where I majored in marine science. And during my time there, I got my first hands-on experience in shark research. I actually did an undergraduate honors thesis where I studied the shark community of our local estuary, Winyah Bay. Specifically, I wanted to look at any changes in the abundance of different shark species over time. But as I was applying to graduate schools, I knew I wanted my research to focus more into fisheries science, and this directly related to our current advisor, Dr. Silikowski's research. So I moved up to Maine and started the master's program at UNE, where now I study how the effects of different fishing practices impacts different fish and shark species in the Gulf of Maine.
Okay, and my name is Hannah. As Tanya said, I grew up in the very landlocked state of Arkansas, and I did decide to stay in my home state to do my undergraduate degree. And I graduated from the University of Arkansas with a degree in biology. While I was there, I got my first taste of research by doing an honors thesis project on algae in streams, which was a really great experience. It got me some hands-on activities with research in the lab. But I knew I was a little bit more interested in the marine biology side of things. That led me to participate in a summer program in Florida where I got to tag sharks and rays for the first time. And that really piqued my interest and got me thinking about how I could pursue this field in the future. So after I graduated, I did an internship at the Bimini Biological Field Station in the Bahamas. It's a world-famous research facility focusing exclusively on sharks and rays. So I got to do a lot of hands-on experience with a lot of different species and kind of got to figure out more of what I wanted to do moving forward. And all of that led me to apply to work with Dr. Silikowski at UNE here in Maine. So I moved up, and we both are in our second year up here working with Dr. Silikowski. So a lot of people ask, why Maine? If you guys have seen Shark Week or anything like that, you probably picture shark research is happening in tropical locations with really clear, shallow water and cool species. But what a lot of people don't know is that the Gulf of Maine is actually a really unique ecosystem that is home to up to 8 species of sharks. One of them is the great white shark, which is probably the most famous and notorious shark species, and it's actually increasing in numbers in the Gulf of Maine as our seal populations continue to increase.
So one of the most common shark species in the Gulf of Maine is the blue shark. And this is actually the most widely distributed shark in the world. It can be found in most oceans, except really in those polar regions. And this is actually one of the most sought after species for its meat and its fins. And that is because of its characteristic and really vibrant blue coloration that's really attractive for meat products.
Spiny dogfish are also really common up here in the Gulf of Maine. This is a pretty small species. They only reach about 3 feet in length. And while that does seem small when you think about big sharks like great whites, actually the majority of shark species are only about 3 feet long. This is a pretty common species, and we like to do a lot of captive research on it since it is so small and easy to handle.
Another shark species that calls the Gulf of Maine home is the thresher shark, which has one of the most unique feeding techniques of any shark species. So that really long caudal or tail fin that you see is actually used kind of as a weapon. So this species will actually whip that tail and strike into a school of fish, which will cause a few of these individuals to be stunned, and then they can easily eat these individuals.
Basket sharks also spend a lot of time in the Gulf of Maine in the summer months. This is actually the second largest shark and actually fish species in the world, second to the whale shark. These guys are actually commonly mistaken with great whites when they're on the surface because they both have those really big dorsal or top fins. But these guys are completely harmless. All they eat is plankton, and so they're called filter.
So next we have another shark that you guys probably recognize. This is the sand tiger shark. And it is often kept in aquariums, and it's really recognizable from those really gnarly snaggle teeth.
Makos are also found in the Gulf of Maine. These guys are really torpedo-shaped, and they're actually the fastest shark species in the world.
And finally, my personal favorite shark species is the porbeagle shark. This is actually a slightly smaller and lesser known cousin of the great white. And it is also much more elusive or harder to catch, which gives it its nickname in our Solikowski lab as the phantom shark.
So out of these 8 species, research in our lab typically focuses on 4 of them, and those are spiny dogfish, great white, blue, and porbeagle sharks. And today we're going to talk to you guys about what it is exactly that we study about these species, how we study it, and how we use that information to better conserve and manage these species. And we'll start with reproduction, which is one of the biggest themes of research in the Solikowski lab, and the specific reason why I moved up here to work with Dr. Solikowski. Reproduction might seem like kind of an odd choice at first, but it's actually one of the most unique and interesting things about this group of fish. For example, some sharks are egg layers, so similar to birds and chickens. A mom will lay an egg. That's what they typically look like. We call them mermaid's purses. And she leaves her egg on the seafloor and doesn't take care of it or do anything. It's on its own. But other shark species actually give live birth, so the babies complete development inside the moms, and they're born looking like miniature adults. But out of these live birthing sharks, the way that the babies complete development inside the mom can vary pretty widely. For example, some shark species just act kind of like incubators to the eggs. So the mom will become pregnant, eggs will form, but she won't lay them out into the environment. She'll keep them inside of her body, which kind of provides a little bit of protection and stability for these babies. But she doesn't really play any major role in their development. And so an example of a species up here that reproduces this way is the spiny dogfish, and we call this yolk sac viviparity. But some moms take this a step further and actually feed their babies while they're inside their uterus. So this is called autophagy, and what happens is the babies will actually learn to eat while they're inside their mom's uterus. She'll take unfertilized eggs from her ovary and deposit them into her uterus, and the babies will eat them. And that's what you see in that big stomach. It's just full of yolky eggs. And actually great whites, porbeagles, makos, and threshers all reproduce in this way. But one species of shark actually takes this another step further. So the sand tiger shark reproduces in what's called adelphophagy, or embryonic cannibalism. So the embryo that grows the fastest will actually eat its younger siblings, and so only 1 or 2 babies are born out of each litter of sand tiger sharks. And then finally, some sharks actually have an umbilical cord and a placental connection to their wing. So it's really similar to mammals, just like us humans. And blue sharks actually reproduce in this way. So like I said, reproduction is really interesting, and I find it absolutely fascinating and worthy of interest all on its own. But reproduction is also one of the most important aspects of biological research for sharks, and that's because of its importance in their conservation. So reproduction fits into what we call the life history characteristics of a species. And these are basically just a set of characteristics that describe how organisms grow, survive, and reproduce, and all of the changes that take place in their body throughout their lifetime. So an example of a life history characteristic would be their lifespan, or how long individuals of a species typically live. It also includes when they go through sexual maturity or when they go through puberty. And it includes characteristics regarding pregnancy. So what is their gestation time, or how long are they pregnant? How many pups do they produce at once, and how often do they become pregnant? Together, all of the life history characteristics of a species kind of determine how susceptible it is to decline. And sharks, in general, share some life history characteristics that make them particularly susceptible to declining numbers as a result of fishing. So for example, a lot of sharks have pretty long lifespans. Many live up to like 20 or 30 years, and the longest living shark species is actually the Greenland shark, and it can live over 450 years old. Sharks also have what we call low reproductive output, so their pregnancies can last anywhere from about 6 months to 2 years, just like the spiny dogfish. And they have relatively few pups at one time, so some species, as I mentioned, only have 1 or 2. Others may have up to about 100, which might seem like a lot of babies to have at once. But when you compare it to something like a bony fish, which can have, hundreds of thousands of eggs at once, it's not very much that they can have. And this is a particular concern for sharks because they're under a lot of threats. Many sharks are targeted for their meat, so people do eat sharks in a lot of places in the world. They're also targeted for their fins. You guys might be familiar with the shark fin trade. Others are caught just for sport because who doesn't want to catch a shark and take a photo with it? And then a lot are caught as bycatch, which Brooke will go into in detail a little bit later. And it's estimated that a quarter of the world's 500 species of shark are at some risk of extinction. So that means that their numbers are going down and they're not able to keep up at a healthy population level due to all of these different threats that they're facing. But by being able to answer some basic reproductive questions, we can start to figure out how best to conserve and manage shark species. For example, if we know when and where reproductive events take place, we can start to identify important habitat for a species that we can potentially protect to give that species a leg up. For example, if we know that most individuals of a species go to a certain area at a certain time of year to mate, we can protect that area at that time to give those individuals a chance to contribute to the next generation. If we know things like how often females give birth or how many pups they produce at once, we can start to estimate their susceptibility to decline. So the longer it takes for them to produce a litter of pups and the fewer pups produced, the more susceptible that species will be to going extinct. Now that you know why we study reproduction, I'll tell you a little bit about how we do this. And we do it using 2 primary methods, which are ultrasound and blood hormone analysis. So the use of ultrasound in shark research is relatively new, but it's based on the exact same technology that's been used on humans for over half a century. So I'm sure you guys are all pretty familiar, but just to give you a refresher, ultrasound works by sending very high frequency sound waves into a shark. And once those sound waves hit some type of barrier, in this case, say a baby shark, some of the sound waves will be reflected back to the ultrasound probe. The probe can then register all of those reflected sound waves and produce an image of what's going on inside the shark. So performing an ultrasound on a shark isn't quite as straightforward as it is on a human because obviously sharks can't talk and they have sharp teeth. But luckily for us, a lot of sharks have a state that we call tonic immobility. So if you rotate them upside down, they go into this really calm state. They're not stressed out. They kind of just continue breathing and everything else shuts down. that's just an example of us giving a spiny dogfish ultrasound. It also, luckily for us, works on the really big species like tiger sharks that we are unable to pull up on a boat or keep in a tank. And sometimes sharks are small enough for us to pull up on the boat when we're out doing research. And so we can just do a quick ultrasound scan while we're doing our other measurements on the shark. Once we take the scan, we have to figure out what the image is telling us. So this is just an example of an ultrasound image of a uterus of a shark that has nothing in it. So this shark was not pregnant. And that's in comparison to something like this, where we can start to see the bodily forms of the sharks. So those two red arrows are pointing to spines or the vertebral columns of two shark pups. This is another example of a face of a spiny dogfish pup. So the arrow points to the very tip of its nose, and if you look a little to the left, you can start to see its mouth. This video shows the head of a spiny dogfish pup as if you're looking straight at it. Sometimes it's hard to see. I think it looks just like the alien face. You can see, I think it's really cute, but it's kind of creepy also. And just because they are so cute, this video is what spiny dogfish pups look like just after birth. We call that the dogfish dance. Sometimes we can actually see these babies moving while they're still in the uterus. So this video shows a tiger shark pup. Well, that video was supposed to show a tiger shark pup opening and closing its mouth as it breathes while it's inside the mom. Unfortunately, it's pink. All right, moving on. As I mentioned, the use of ultrasound in shark research is relatively new. So we're constantly learning new ways to apply this technology to our research. And as I mentioned, not all shark species reproduce in the same way. So we don't always know what an ultrasound image will look like for a particular species until we can verify that image in real life. So recently, Brooke and I had the unique opportunity to ultrasound a pregnant poor beetle shark for the first time. So a fisherman caught this shark and brought it in, and luckily our lab was called before the shark was cut open because we don't like to let anything go to waste. You can always get really good data even if the shark is dead. So we didn't really know what to expect. I hope this video works. So we took an ultrasound. But you can kind of see in the center, it's this video, there's this weird form going on. We thought it might be something like a pup, but we weren't really sure. But when the shark was cut open and dissected, this is what we saw. It was pregnant with 4 pups. They looked super odd. And it was actually pretty much exactly what we were seeing on the ultrasound. So we did verify what a poor beetle embryo looks like. So you might remember that I mentioned poor beetles reproduce by using euphagy. So that's when the mom takes eggs out of her ovary and puts them in her uterus to feed her pups. And this is actually the ovary of that mom. It weighed like 10 pounds. And every single yellow dot is an unfertilized egg that she's going to use to feed her pups. So they have these really sharp teeth that helps them pierce those eggs and get the nutrients. So they're learning to hunt still while they're in utero, which is really cool. Briefly, before I wrap up reproduction, I did mention to use another method called blood hormone analysis to study shark reproduction. It's a lot less exciting and fun compared to ultrasounding sharks. So I'm not going to go into a lot of detail, but it is a really important tool because not all researchers have a portable waterproof ultrasound that they can take out on boats. But almost anyone can take a quick blood sample from a shark, and with that blood sample, we can do some experiments in the lab and look at the amount of different hormones present in that sample to get an idea of where a shark is in its reproductive cycle. So for example, a hormone that we look at a lot is testosterone. If we look at the amount of testosterone in male shark blood, we can get an idea of whether that shark is about to mate or whether it's already mated for that year. So with that, I'll go ahead and wrap it up and give it to Brooke to talk about the next part.
Okay guys, so now I'm going to switch gears into one of our other overarching research goals or areas in our lab, which is fisheries bycatch. So I'm going to start with a bit of background information in case anyone is unfamiliar. But a fishery can be defined by a geographical area, the gear type that is used, and by the species that is targeted, which is typically what you would buy at the supermarket or a restaurant. But fisheries don't only catch the target species that they are looking for. They can also catch a wide variety of different organisms, which leads us to the topic of bycatch. So bycatch is any non-targeted organism that is captured in fishing gear and then is discarded alive. Now, a lot of times shark species are actually targeted in fisheries, but today I'm just going to focus on those portions of sharks that are actually discarded alive. Now, there are hundreds of different fisheries across the globe, and bycatch occurs in each and every one of them to a certain extent. And this is typically because fisheries oftentimes are non-selective, which means it's really difficult for a fisherman to go out and only attract, only find, and only catch the type of species it wanted. So I'm going to start by giving you guys a little insight into bycatch that we see in 4 fisheries in the Gulf of Maine, starting with the Atlantic sea scallop dredge fishery. This fishery, of course, hence its name, targets sea scallops. And the scallop dredge fishery uses a large, heavy metal bag that is dragged across the seafloor, which is really good at catching sea scallops that live on the bottom, but is also going to catch anything that's in the path of the dredge bag on the bottom. And this can often include invertebrates such as sea stars, but a lot of times it catches fish species, including sharks. I have photos of 2 shark individuals we caught in the scallop dredge fishery as bycatch. On the right is the very interesting looking angel shark, and on the left is the spiny dogfish. So another example of a fishery in New England is the gillnet fishery. A gillnet is basically a large mesh net that is stretched across the water column and is actually left sitting in the ocean for several hours or up to a couple of days. And this fishery targets what are called monkfish, or you guys may have heard of it called goosefish. But anything that might swim into that gillnet will actually get entangled and captured. And sharks are, since they're such good swimmers, they actually are caught as bycatch in this fishery quite often. And one of the main bycatch species in the Gulf of Maine gillnet fishery is once again the spiny dogfish. Another really popular and economically important fishery in our area is the otter trawl fishery. This is a large rope bag that is towed behind the fishing vessel, and it is used to catch a variety of different ground fish species that you guys might like to eat, such as the haddock shown here, as well as pollock or Atlantic cod. But similar to the scallop dredge fishery, this otter trawl will catch anything that's swimming in the path of the bag. And once again, shark bycatch occurs quite often in this fishery. On the left-hand photo, every gray figure that you see in that photo is a spiny dogfish. You can see they're caught quite often in this fishery. And on the right is a pretty large porbeagle shark. And the last fishery in the Gulf of Maine that I'm going to use as an example is the longline fishery. Now, a longline is pretty much exactly what it sounds like. It's a really long line that can stretch up to miles in the ocean. And attached to it are thousands of hooks with bait. And this is used to catch large fish species such as tunas or swordfish, but anything that might be swimming in the water that would be attracted to and try to eat this fish or squid bait that is used can be captured. This can even include things like marine diving birds marine mammals, sea turtles, and oftentimes sharks. The three main bycatch shark species in this fishery are the blue shark, the mako shark, and the porbeagle shark. Now, like I said, all of this shark bycatch is released alive, which might sound really great for shark populations, but a capture in a fishery is actually a really stressful process for sharks. And I'm kind of going to walk you guys through all of the different stressors that a shark experiences during capture in a fishery. So first and foremost, sharks are subjected to interactions with the fishing gear itself. And this can cause a wide range of different injuries depending on the type of gear. So hook and line fisheries, such as the longline fishery, will cause hooking wounds, whereas net fisheries such as the gillnet, the otter trawl, or the scallop dredge fishery will cause abrasions due to entanglements in those nets. Then upon retrieval of the fishing gear, sharks are subjected to rapid changes in depth or pressure, as well as temperature. And these can cause a wide range of internal stress. And they can also be subjected to really long capture durations. In some instances, sharks are left on a line or in a net, struggling for up to 12 hours. Once the shark reaches the surface, it experiences a bunch of environmental conditions that it's not used to, such as changes in air temperature, which is typically different than water temperature, as well as direct exposure to sunlight and rough sea surface conditions. So if any of you guys have been out on a boat on a really rough day, you know being seasick is absolutely no fun at all, and it's not fun for sharks either. Once the sharks are brought on the fishing vessel, they can be handled by fishermen. Some of these techniques can include a shark metal gaff, which can cause further injuries to the animal. And they are also exposed to air. So of course, sharks breathe water. They can only get oxygen from water. They can't get oxygen from air. So you can kind of think of this experience of a shark out of water, similar to when humans are held underwater for too long. Finally, the shark is released back into the ocean, and if you guys thought your upcoming exam was stressful, I promise it's nothing compared to what a shark goes through during capture in a fishery. So because of all this stress, shark bycatch are more susceptible to disease, in some cases predation by larger animals, and ultimately mortality or death. And so one of the largest questions that our lab seeks to answer is, what is the fate of discarded shark bycatch? Are they surviving or are they dying? And there are several reasons why we're interested in studying shark bycatch mortality. One of them is that if survival of these species is low, it can cause a variety of different negative impacts. For example, if all of the sharks that are being thrown back are dying, it's a substantial waste of resources. All of those individuals could be used for food or other products. Additionally, if sharks are caught in large numbers and are not surviving, this can cause substantial population declines. And this is particularly important for shark species because of all of those life history characteristics that Hannah mentioned earlier, such as late ages at maturity or low reproductive output. All of these factors make sharks even more vulnerable to overfishing. And again, as Hannah mentioned earlier, 1/4 of all shark species are threatened with extinction, which just highlights our need to study their mortality due to fishing. And finally, understanding the post-release mortality of bycatch is important for management because it can be incorporated into the catch limits of a species or how many fishermen are allowed to catch or discard. And this can aid in the sustainable management and conservation of sharks. So now that we know what bycatch is and why we're interested in studying it, I'm going to go over how we study the post-release mortality of sharks, and we do so using satellite tags, specifically pop-off satellite archival tags. This type of tag is attached to the shark directly below the dorsal or the top fin, as you can see in the photo on the right. And this doesn't add any substantial harm to the shark. It's similar to getting an ear pierced. But in this video, if it will play, you can see that I've attached a pop-off tag to this juvenile porbeagle shark. And we will also take several measurements as well as look at the condition of the animal. So we'll look for any external injuries, or we'll look at how lively the shark is after release to kind of get a prediction as to whether or not the individual will survive. Excuse me. So once the shark is released with its new jewelry, the pop-off tag will collect fine-scale data on the temperature and the depth, more importantly, the depth profile of the shark. And then after 28 days, or if the shark dies, the tag will be released from the shark. It will float up to the surface. And all of that stored data will be sent to a satellite and then back to our lab for us to analyze it. And from this data, we can estimate whether or not that individual survived or died due to capture. So the example that you will see here is the first 3 hours after capture, the depth profile of a juvenile porbeagle shark. As you can see, this individual moved up and down in the water column and never stayed at one depth for too long. And this indicates to us that this shark was able to recover and survive the capture and handling process. In comparison, this is the depth profile we got for 3 hours after capture of another porbeagle shark. This individual slowly sank to the seafloor at about 250 meters and then stayed at constant depth for the rest of its tag duration for 4 days. And from this profile, we can see that this individual, unfortunately, was not able to recover from the capture and handling process. So once we've collected data from enough individuals, we can get an overall estimate of the post-release mortality rate of a shark species in a given fishery. This information can be provided to management entities. It can be incorporated into the catch limits of the species. And we can also provide fishermen and management with recommendations for best fishing practices. For example, we can recommend specific gear configurations that would minimize the injury of the animal. We can recommend reductions in the capture or fight durations, and this would reduce the amount of time an individual struggles in a net or on a line. And finally, we can recommend reductions in the amount of time individuals are exposed to air. So in the case of hook and line fisheries, such as the long line fishery, we can recommend that fishermen cut the line close to the hook so that the shark doesn't even have to be brought out of the water at all. Or in the case of net fisheries, where the catch has to be dumped on deck, we can recommend that the shark bycatch be released immediately back into the water before any other handling or sorting processes begin. Now, so all of these recommendations can be used to help sustainably manage and conserve the bycatch species and ultimately help improve the survival rates. So now we're going to move on to our last overarching research area that we will talk about today, which is movement and habitat use. So this is how we look at when and why a species is going to a particular habitat, and eventually we want to understand why they're using these locations. And we do so using satellite tags. This is a different kind of tag than you saw before. It's called a fin mount tag. It's attached directly to the dorsal fin of the shark. And once again, this is similar to an ear piercing. It doesn't cause substantial injury. And in this video, you will see that we've attached a fin mount tag to the dorsal fin of an adult porbeagle shark. So we will release this porbeagle shark, send it on its way to do its shark business, and every time that shark comes up to the water's surface, that fin mount tag will send a location transmission to a satellite, and it's similar to using a GPS in your phone. We get real-time locations from that shark.
So movement data of sharks is important for a variety of reasons, but we typically use it in the context of our other topics of research. And that's so that we can get a more comprehensive or holistic view of the animals that we're studying. So for example, we often use movement data in the context of life history or reproduction. This might include looking at whether the movements of juvenile or baby sharks differ from those of adults. That's because these two life stages typically inhabit very different areas because sharks are pretty cannibalistic. So a big shark will take any chance it can get to eat a smaller shark, even if it's the exact same species. We can also look at whether pregnant females use different areas compared to non-pregnant females, because that can help us get an idea of where reproductive events take place, which, as I mentioned earlier, can really help us conserve these species. So this is an example of the movements of a pregnant porbeagle shark that we tagged. So we ultrasounded it, determined that she was pregnant, put a tag on her, and sent her on her way. And as you can see, she moved around at the surface quite a bit, but she stayed pretty close to shore. Very long range movements. And this is in contrast to a porbeagle shark that we tagged that was not pregnant. So this map is a lot more zoomed out, and you can see that this individual goes much further from shore and a lot further south. It's all the way down to New Jersey, I think. So when you look at the movements of these two individuals side by side, you can see that the pregnant shark, which is shown in purple, occupied a very different habitat than the non-pregnant shark shown in green. So this might indicate to us that that pregnant individual is staying close to shore for a very particular reason, maybe so that she can give birth to her pups in a protected coastal habitat.
In addition to studying movement and habitat use in a reproductive context, we can also use it to study in the fisheries context. So we can look at what location a particular species is using and when, and we can compare that to when fishing is occurring in that habitat to gauge any overlaps and see how vulnerable that species may be to capture in different fisheries. So for example, this map shows the summer movements of 5 juvenile porbeagle sharks that we tagged right off of UNE, which you can see in yellow. So as you guys can see from this map, all these juvenile porbeagles are all aggregating really close to shore, maybe a protected area for them. But we also know that tuna fishing is really popular in the summer months right off our coast. And this may indicate to us that these juvenile sharks may be vulnerable to capture in this fishery. And in fact, that is exactly what we saw. 2 out of these 5 individuals were actually recaptured during the tuna fishing season, and this further suggests that this species may be vulnerable. So we can use this information to provide to management recommendations for possibly altering the location or timing of the tuna fishing season so that we can reduce the impact on this vulnerable species and life stage. So overall, all of the research that we do at the Solokowski Shark and Fish Research Lab is very interconnected, where aspects of our reproduction, bycatch, and movement studies are all integrated to reach our ultimate goal. which is to create more effective management of shark species. And by aiding in shark species management, we are helping to maintain healthy ocean ecosystems and a healthy Earth overall.
That's all we have. We're happy to take any questions you guys have today. You can also feel free to reach out to us at our lab email, which is shown up here. You can also follow our lab on Instagram. We post a lot of regular updates on what we're doing in the field, as well as in the lab. The lab name, Bycatch Lab.
Two questions that I got, so we'll just take a couple minutes. So one was, are there shark species endangered in the Gulf of Maine? And the second was, with a pop-off tracker, how do you know when the shark died?
So a lot of the shark species in the Gulf of Maine are not endangered, such as the spiny dogfish and blue shark. The horned beagle is actually considered endangered by the International Union for the Conservation of Nature, but not by the U.S. government. So it's not protected as an endangered species, even though its numbers are suggesting that it might be.
And the second question was, how do we tell if the shark died using the pop-off tag? So the fine scale depth data that we get is the way that we determine whether or not the shark lived or died. We expect that a living shark is going to be moving, making large depth changes as it's searching for food, pretty much searching for food. It's a lot of what they do. But a shark that dies is actually negatively buoyant. So that means that shark is not going to float, it's actually going to sink. And once that shark sinks, it will reach the sea floor and kind of stay there. So it will stay at that depth for the rest of the tag duration. And from that, we can determine that that shark, unfortunately, did not survive.
Does anybody else have questions? I'll look. Show and tell, and then I'm not sure who you are, and then you.
I have two questions. My first question is, how do they get the umbilical cord off the mom and the child?
It just breaks.
Huh?
The umbilical cord just breaks when they're born.
Oh, and then I have another question.
I can ask it for you. Someone asked me how the sharks actually become impregnated.
So male sharks have what's called claspers. They're modified pelvic fins, and it's internal fertilization similar to eagles.
Yeah.
What was your question? Okay, hold up, guys. I know we're almost done. So she's asking, once the tag falls off, how do you get it back on? How do you collect them back?
That's a really good question, and the really unique thing about these tags is, remember they're called pop-off archival tags, and the archival aspect of it means that all of that data is stored in the tag for the rest of the deployment duration, and then once it pops to the surface, it actually, via satellite, transmits all that data back to our computer. So we actually get all of that data as soon as it pops to the surface through that satellite. If we do recover the tag, which we can get location after it pops up, so if it washes up on a beach, we can potentially find it, and that just adds, we get more data if we do find the tag, but it's not required for us to get that information. Does that make sense?
Friendly when you hold them.
So sharks are wild animals. I don't recommend handling sharks if you are not trained to do so. We do it with all of our team's safety and the shark safety in mind. So we're able to handle them, to restrain them very carefully. They don't want to hurt us, but they are scared when we catch them, so they might be trying to thrash it out and bite. It's not too hard to put the tag in. sharks are made of cartilage, so it's kind of like your ear and your nose. So it's kind of like piercing them, how we get our ears pierced.
All right, I think we have one more question in the back, and then we'll wrap it up. Yeah.
If you get the pop-up thing, if you can find it, can you use it again? Or
Can you use the pop-up tracker again if it pops up?
Yes, you can, which is another really great thing if you are able to find it. You can get more data from it from another shark.
Our number and email is on the tab, so when people find them, they often call and they really report. So it's a little incentive for someone to find their tags to get back to us.
All right, let's give another round of applause for our guests.